Click for IB Biology. We're going to be looking at what is called a point mutation and the way this affects the protein which is manufactured in protein synthesis. The example that we're going to be using is the Sickle mutation for hemoglobin, and we'll have a look at how the mutation can change the shape and the function of the protein. There's a blank PDF of this presentation down in the description. You can annotate that with your own notes. Quick quizzes, 10 questions and answers. Make sure you can do those. And the channel link has an AI prompt which you can cut and paste into ChatGPT or one of the other chatbots, and it will produce an individualized multiple choice assessment for you. So let's look at the idea of point mutation. Now, point mutation means that only one base, so the point that we're discussing is that a base is changed. Now, the kind of things that might change a base in a gene are called which are called mutations, are mutagens. The kind of things that cause mutation are chemicals, radiation, of course, and viruses. And then that's our starting list. So what we're interested in here is, how does a base change the protein? So we have here a genetic messenger RNA sequence, CGC, GUU, UCC, and this codes for the amino acid sequence arginine, valine, serine. So this is the original. Now, point mutations can be of the type known as substitution. In other words, we're going to swap one of the bases. And in this particular case, we're going to swap for the G, we're going to swap in an A. So notice that this is occurring here. And so our base sequence is now going to read CGC, AUU, UCC. That means that we only have one base change, but look what this has done to our protein sequence. So arginine, ILE, alanine, serine. So we have one amino acid change in our sequence here. So just one amino acid by a substitution. Now that substitution can occur anywhere in the gene. This is just one that we're using to illustrate the purposes. So that's one amino acid change. The deletion of a base, however, has a much more drastic effect. So if we delete G, then our code will now read CGC, but the G's gone, remember. So now we shift our code on to UUU, CC, and the next one down here must have been an A. We didn't know that, but it is. And this is called frameshifting. So the way in which the messenger RNA codons are read by the ribosome has frameshifted. And this has created an entirely different. So we've got arginine, then we have phenylalanine and proline here like this. And so we've got two changes. So for one base change, we've ended up with two amino acid changes. So this is a very big change. So deletions often result in very big changes to the protein sequence. And this is called the frameshift effect. The alternative to substitution and deletion is to insert, to put something in. And so we're going to add adenine to our code in the same position. And so now it reads CGC. Here's our new adenine. And so this shifts the code reading to the right. So we're going to go CGC, AGU, UUC, which gives us arginine, serine, and phenylalanine like this, which again is an example of frame shifting and has affected the entire protein. So this one here affects one amino acid, but the other ones affect many amino acids. So you can imagine the likelihood of these proteins still being functional and working well is remote. Let's take a look at an example of a point mutation that affects the structure of a protein, and that protein is hemoglobin, which often is abbreviated to HB for the protein. So we have the normal gene here, which is HBA, and then we have another one which is called the Sickle version, which is HBS. And you can see here that in DNA we have GAG at the codon, and complementary is CTC. And when transcribed, that would give us GAG. So this is the antisense which has been transcribed. And so we get GAG, which translates to glutamic acid. And this would be what we would find in normal. So that would, let's call that normal, for normal hemoglobin. Now for a single base change at this position here, we're going to change that to T. So A is turned into T, so that's a substitution. And so now, when transcribed, our codon has been changed to GUG, and this translates to the amino acid valine. And so for one amino acid change, so one base change, has caused one amino acid change in this hemoglobin protein, and this has the effect, these details are not required at this point in the syllabus, but it's very interesting to think about this, that the mutation has changed the protein in hemoglobin, and this causes the shape of the cell to change, and this cell shape, which is known as the sickle shape, can cause all sorts of physiological damage. Perhaps the most famous version of this is infarction or blocking of the capillaries within the kidney, and that ends up causing damage and kidney damage, which is unfortunate for someone with this single point mutation which changed the protein hemoglobin. The next video is coming up here. That's a higher level video. The channel subscription's there, and the playlist continues. A few more videos to cover in this topic of protein synthesis.